So, turn this on again. Does it look like it's on? I thought I turned it on, but there it is. I did turn it on. I did, and I will hover over by it and speak loudly. But I didn't wear a pocket today. There we go. So, you don't have this presentation, and that's intentional. Because I want to just think about some things. And so I'm just going to take about 20 minutes and talk about phytoplankton functional types and particle size distributions in the ocean, so that you can think about that while Jeremy gives you a talk on particle size distributions and functional types in the oceans from modeling. But I want you to think about what's really in the ocean. Okay, so I spend most of my academic time teaching students about oceanography. And so I just want to kind of get everybody onto the same thought process so that when you think about inversions and you think about the results that we get for these types of reflectance modeling, that you can put it in the perspective of what's really going on. And so I'm just going to throw this image up, and this is sort of a mission average or an annual average satellite chlorophyll derived from an ocean color. This is, I believe this one is Modus. And so you see these really beautiful patterns, right? You've seen these before a number of times in the last 3 weeks. But I think it's really important to delve in and really look at the spatial patterns of phytoplankton across the global ocean. And so what I want to think about for the next 20 minutes is what drives the distribution of chlorophyll, because that's really what we're measuring is chlorophyll, remember, or some greenness index that we get chlorophyll from. And then step away from that and say, okay, well, let's think about that being biomass, some proxy for biomass, and what is driving the concentration of phytoplankton and the distribution in the ocean. Okay, so what are some important factors in the ocean that drive this pattern?
Nutrient availability.
Ryan, what'd you say? Oh, no, you didn't say it. Did you say something? Lectin nutrients. Hmm? Currents?
Temperature,
Hmm? Temperature?
maybe.
Hmm?
Proximity to land.
So what would land do? Is it affecting one of these? Okay. Yeah. Mixing?
Prey field?
Hmm?
Prey field?
Prey field?
The predators?
Predators, yes.
Thank God, somebody think about somebody.
Yeah. Any other lost terms that might be important? Big particles do it, small particles don't. Yeah. Okay, so if you think of any others, let me know. But when you start to look at these major drivers, you want to think about what's going to be a primary driver and what's going to be a secondary driver. But when you add up the effects of all of these and you scale them correctly, we should be able to predict this. So what do you think is a primary driver? In this order, it's always light and nutrients. I very rarely hear nutrients and light. It's really funny. So, light. So, what does light look like over the globe? Shaped like this. Like this? More or less. So you mean that it's less in the poles and more at the equator? Yeah, exactly. And so I actually tried to pull up something, top of the atmosphere, net solar radiation. This is an annual mean. And you can see that it's, over the year, it's definitely lower in the polar regions and definitely higher in the equatorial regions. And, you know, the Earth, the relationship of the Earth on its tilt relative to the sun kind of leads you to some higher bands. But that's essentially what's coming into the top of the atmosphere. And I honestly don't understand why it's not completely uniform. I think it has to do with the way that they've measured it. But essentially, it looks like this in terms of latitude. Latitude. Okay. So is that really what hits the ocean? No. So what hits the ocean? What's transmitted through the atmosphere? And so it looks something more like this. Okay. That's the incident solar radiation. That's an annual average over a number of years, 20 years, 10 years. So it looks like this, except you see some different patterns. So why are you seeing different patterns? Clouds, yeah. So you're getting a lot more solar radiation in the southern hemisphere than in the northern hemisphere, right?
Do you know there are other gases, aerosol, and ice?
Yeah, there could be different gases, different aerosols, different cloud covers. And if you look just at the cloud cover, that gives you a lot of, you know, sort of this second order effect. Like you have all this light coming in, but then you have this modification by clouds. And the distribution of clouds is related to a lot of different things. And you could go and identify the factors that are responsible for really high cloudiness versus low cloudiness. You know, you can identify places where we have rainforests, places where we have deserts. But that's essentially determining the amount of light that is reaching the surface of the ocean. Okay. So if we go back and we look, does that explain the distribution of phytoplankton in the surface ocean? No, it doesn't. Because I want you to keep that in mind and go back and look at this. So is light determining phytoplankton in the ocean? Not at the surface, not the surface global distribution. When you look at the dynamic range from the clearest waters to the most turbid, you can't explain that pattern with the spatial pattern of light. So, not global, and not surface. We know light is important, right? But not on this scale. Is that something that you had expected? Or do you think?
Is this also an at-home?
Yeah. Yep. So let's think about nutrients. Right. So what do nutrients look like in the ocean? Huh?
That's tougher.
That is tougher, right? Because basically we measure what's left over. Right? So you want to think about what would be there if the phytoplankton weren't there. So what determines the nutrient distribution in the ocean? If you could remove the phytoplankton and just look at the physical sources of nutrients and the regenerative sources of nutrients, where do nutrients come from? Upwelling. Upwelling of what? Huh?
Water?
Yeah, upwelling of water, but what water? Deep water. So, you know, in the ocean, you have these permanent pycnoclines, and you have all this deep water that's really high in nutrients. And so there are places where you can get those nutrients into the surface. So where do we tend to see that happen? Where? The equator. Huh. Cool. The what? Eastern boundary currents, upwelling currents. Ooh, look at this. Ooh. Cool. Where else? The poles. So we have upwelling in the poles. So are you talking about the poles, or are you talking like, so the pole down here doesn't have an ocean, right? The pole up here is covered by, ice most of the year and clouds, and also the polar orbiter can't get up there. But, would we have, so we have this high, I don't have my pointer thing. We have these high regions in the subpolar regions, right? And the subpolar regions down here. So those are high regions, and then we have these low in the subtropics. So what's happening in those regions? What- What's stratified and mixed?
Like in high latitudes, you see the stratification is not strong.
All the time?
More in the winter.
Yeah, so in the high latitudes in the winter, it's
still the difference between the temperature of the deep water and compared to mid latitudes, it's not as strong. Easier.
What were you going to say?
Just higher wind speed.
There's gradations in wind speed, yeah. Yeah, there's Ekman pumping. So if you look at the surface nitrate, you can see that it's really high down here. It's high at the equator where we have upwelling. There's spots of high along the coast and high in the subpolar regions. And you can go back to deriving from sea level pressure, right? Atmospheric sea level pressure. And from that you can derive winds, right? And from winds you can derive geostrophic balance. And you can end up with global sea surface height differentiation, which says that in these subtropic regions the sea surface height is higher, and in the subpolar regions the sea surface height is lower, right? Because of Ekman transport. And so what that means is that there are places where you get transport, convergent transport, which piles up the sea surface. But what that does is it pushes down on the picnic line and depresses the picnic line, isolating that nutrient-rich water deeper. And that's what occurs in the subtropics. And by the same token, you can have places where divergence away lowers the sea surface and also causes a bowing up of the picnic line. And so now the nutrients are closer on a large scale. And yes, seasonally, this also, you have the seasonal breakdown, which occurs in the subpolar regions. But this is what's really driving the global distribution of nutrients.
But would you say that in high latitudes, nutrients are driving the chlorophyll concentration?
Well, I'm going to ask you that.
Because, so I haven't looked a lot of profiles of nutrients, but would they really be zero on the surface? Is that limiting for the phytoplankton?
See if I have it here. Well, so this is what we measure.
So it doesn't look.
So they're very low in the subtropical gyres, right? And there's a couple of reasons why they're low. They're low because of this. Because if you had if you could start the ocean without biology, and you can drive it with large-scale physics, and you end up with this thick, warm surface layer, but you have and very far, very deep, like 100, 200 meters, isolated picnic line, and you had any phytoplankton in there at all, what's going to happen to the phytoplankton that are in this layer? What are they going to do to whatever nutrients were there? Consume it. Now, what's the regeneration rate of nutrients in these waters? It's very strong stratification. It's deep. And so there's not a lot of regeneration. And so you measure zero because it's been used up, and the rate at which there's nutrient replenishment And the phytoplankton that are there are utilizing it very quickly.
But
Okay.
this is not what you see in the southern ocean.
No, there's other things going on in the Southern Ocean, right? Maybe other things are limiting. But here what you're seeing is a replenishment, very high replenishment because it's a divergent zone around the equator, around the Antarctica. So then my question is, all right, well, we see that it's low in the subtropics, we see that it's high in the subpolar regions, and that seems to look a lot like this. Then my question is, who's living there? Okay. There are phytoplankton living here, not many of them. So who best to take advantage of those situations, those conditions? Okay. And so what I want you to think about is phytoplankton size. So this relates back to what Emmanuel was talking about. And we're assuming spheres because the math is easy. And I want you to think about the surface to volume ratio. Okay. So let's consider a couple of cells that maybe have a 1 micron diameter, a 2, and a 4. Okay. So you can calculate the surface area in microns squared. And you can calculate the volume. What's the equation for the volume for a sphere? 4/3 pi r cubed, right? So you can calculate that for each one of these. And then you can take the ratio of the surface to the volume. And this is exactly what Emmanuel said, that spheres have the highest surface to volume ratio of any shape. And of course it changes. Pardon? Lowest, lowest. But it really depends on the size of the cells, right? So you can see that these small cells have a very large surface to volume ratio compared to these big cells. So why does that matter for nutrients? So they absorb nutrients through their surface area. Okay, some have active sites, but not all. But it's the surface through which they're collecting nutrients for growth. Right? So which one of these is going to do best in low nutrient waters?
Small cells.
The small cells. Right? And as it turns out, small cells, because they're more efficient in these dilute nutrient environments, we tend to find them in low nutrient waters. Large cells have a lot of advantages. Number one, they have a big volume in which they can store nutrients. So they tend to do well in nutrient patchy environments like upwelling zones, where there's because upwelling isn't constant. It comes in pulses. So it pulses up, you get lots of nutrients, the cells can suck it in, they can store the nitrate and break down to ammonia or whatever storage product, and then they can chew on it for a while. Right? And small cells don't have that advantage. And so we tend to find large cells in places where we have high nutrient concentrations. They also have, take advantage of high growth rates. And so we can look at just these physical constraints and make some predictions about who we are going to find where. And so when you go back and you look at the nutrient environment where these look like the subtropical regions, what we call the oligotrophic, low nutrients. And these are the subpolar regions. And sometimes we call these eutrophic, or higher nutrient environments. We would make predictions that we would find small cells in the subtropical gyres and dominance by big cells in the subpolar gyres. So, how many people have gone out and measured size distribution in the ocean of phytoplankton? Besides Emmanuel. Okay, well, when people go out, this is what they tend to find. They can collect samples, they can do size fractionation, they can do pigmentation, and it's consistent with the small cell species. And so this is something that Jeremy's probably going to show you, and this is from a paper by Tiho Kostadinov, who took this class a long time ago. And he did an inversion, which Jeremy's going to talk about, where he can predict the slope of the backscattering and use that to derive the size spectrum of phytoplankton. And so what he found was this mission mean, September through December, 10 years of chlorophyll. He also had a mission mean of the slope of the size distribution. And what do you notice? So when this is close to red, it's a very large coefficient, a very steep size distribution, domination by small cells. When the slope is flatter of the size distribution, there's more large particles, steeper, flatter, right? So tell me what you see when you look at the patterns between the biomass of phytoplankton and the size slope of particulate matter associated with them. Catherine, what do you see?
High biomass means small particles.
High biomass, so the subtropical or our subpolar regions and maybe the equator. And what?
Oh wait, sorry, I can't read that.
So this is chlorophyll.
Yeah.
And this is the slope of the size distribution where the blue, purple colors are large particles and the orange and red are small particles.
So low biomass means small particles?
Low biomass means small particles, and high biomass means large particles. Okay. Does that make sense with what we have over here? Is that what you would predict? Low biomass, oligotrophic, low nutrients, small cells, high nutrients, high biomass. Okay. So what do you say? Like, wow, that's awesome. But it kind of looks like a mirror image, right? And so then I asked myself, like, does that mean that every time I measure a low chlorophyll concentration, I always have small cells, Andre?
No, indeed, bigger cells with less chlorophyll.
You can have big cells but low chlorophyll biomass, right? And every time I measure high biomass, do I have large cells? No, you don't. And so I think what's really critical is to think about dynamic range and to think about the first order predictor in the ocean is chlorophyll, right? This drives the dynamic range from 0.01 to 100. And over the globe, we see a 4-order dynamic range in the concentration of phytoplankton. We also see a huge dynamic range in the size of the particles. We also see a huge dynamic range in backscattering. We see a huge dynamic range in beam attenuation. We see a huge dynamic range in any of the properties you want to think about. And so over the globe, global patterns tend to co-vary. And Emmanuel asked a question last week, which was, does your model give you a better prediction than does the relationship to chlorophyll? But if chlorophyll's driving the global distribution, then most things that have a global distribution will look just like chlorophyll or the inverse of it. So then the question to think about as you work forward in your own models, as you review papers, is, but where might those exceptions be? And if your model can predict the exception, the low biomass big cells, the high biomass small cells, then you know that you have a model that is useful for predicting things in the future, predicting change. Okay? But if you want an answer like where the small cells going to be, this gives you a pretty good answer of where you are likely to find small cells. Okay? So I just want to show you, because I have minus 5 minutes. I want to show you an exception. This is the North Pacific. This is the seasonal chlorophyll from Giovanni. You actually measure lower chlorophyll in the winter, depending upon where you select your site. But ask Emmanuel what kind of cells he found in November in the North Atlantic. Did you see diatoms? See a lot of big diatoms, okay? So the question is, when you go back to an absorption model like Anique Rocco's, which I'm a huge fan of because it allows you to scale the specific absorption coefficient to small cells versus large cells, small cells being much less packaged, and she's parameterized this as a function of chlorophyll concentration because at low chlorophyll concentration, I'm in the oligotrophic gyre, I have small cells, and they're unpackaged. So this gives me a nice absorption spectrum for those. And at high chlorophyll, I'm likely over here, and I have big diatoms, and they're packaged, and the absorption efficiency is low. When you look at the spectrum, you see this is the low chlorophyll water, and this is the high chlorophyll water. But if you measure the absorption spectrum in the North Atlantic in the wintertime, and you have diatoms, your absorption spectrum is not going to look like this. It's going to look like this. It's just going to be a low concentration. So again, be careful. Over the globe, this is a great model to predict this level of variability. But over a seasonal cycle, maybe not. So think about that. Yeah.
Does the relationship holds for like, it's just surface or anything, like even chlorophyll on the subsurface piece?
Go look at where the data were collected. It's your assignment. The other example I want to show you really quickly.
It's an excellent question. because,
Yeah. I mean, I know where it is. I want you to go and look.
a lot
Yeah.
of-
Don't answer the
Yeah,
question.
No, I'm not answering.
I remember
A
the.
lot of the relationship, case one relationship were derived from surface water. And people have applied them to depth without ever checking
Yeah.
where these relationships are.
Yeah.
So really important, whatever model you use, before you use it, make sure you know what assumptions have been, but what data is used to derive it.
Brown tides in Long Island. A student of mine went there to measure. This is a bloom of something called Areococcus anthrophogeferens. It's a tiny, like 2 to 3 micron cell that turns the water brown. She measured 2 different bays, West Neck Bay and Quantock Bay. Nothing was going on in West Neck Bay. The bloom was in Quantock Bay. You can see it in the absorption and the attenuation and the chlorophyll and the concentration of cells. And nothing was going on here. Apparently, there was no biomass change over the 2 weeks. She measured the slope of the beam attenuation coefficient and found that there was no change in the slope of beam C, which means no change in size distribution during this entire bloom. When she looked over here, she saw huge changes in the slope and actually was able to find, doing a bunch of other types of measurements, that there were huge changes in the ecosystem structure here. But the biomass was constant and low. And there was no change in the ecosystem structure here, but you had a huge bloom. So it's a really fascinating paper, which talks about like what bulk properties mean. But she tried to use this model to do some measurements. But you can imagine that if you had chlorophyll of 40 and you plug that into this model, you get a very, very strange absorption spectrum. Whereas the absorption spectrum of these cells looked like the oligotrophic. Okay. So one thing that I recommend is taking this model and pushing it to the lowest end chlorophyll below which it was characterized for, and above which it is characterized. Because then you see how the dependencies really depend upon, like, it's only good between 0.1 and 10, but if you take it to 30 or 40, you get really strange absorption spectra. And so then you know what its sort of spectral dependencies are, so it's just a good thing to do. Okay. And then I think the last one was a Procentrum micans bloom, almost spheres, right? Changed the water off of Bigelow Lab in 1 day. And Stacy measured a size distribution with an instrument called a Galai. And it was measuring, so this is size, and she was trying to do the right concentration of cells per cubic meter per meter interval versus diameter in meters. I'm not sure why she didn't convert things to something more simple, but she was calling Emmanuel every day to do that. But I would just have you note that, you know, what the slope that you measure may not tell you anything about the population of your bloom. And so when he showed those other models of how to better predict size slope, I think looking at that is really important. So with that, I'm turning it over to Jeremy. So be thinking about global distributions of biomass, which are controlled by nutrients, and global distributions of cell size, which might be controlled by nutrients. And light is important, but when is light important? Is it important at the surface? Light is important at depth, right? You need light when you want to know the distribution of depth. Good. Okay.
So, the difference, another really important issue with light